Internet, so we'll go ahead and get started here. Harry, are we live yet, or just about? Okay, we're live. Good evening, everyone. My name is Mark Broman. I'm the Executive Director of the Wachiska Audubon. I want to welcome everybody tonight here in person, and then those of you online at home. Someone reminded me I wanted to make an announcement. Don Pepperell passed away on Monday, and the obituary was in the Lincoln Journal-Star on Tuesday. Don was our pro bono legal counsel for 29 years, plus his building is where our home is. The Wachiska office and the Nebraska Wildlife Federation and some other folks are in his building at 46th and Calvert. So Don passed away, and he very generously, his family has made the memorials to Wachiska. So that was awful nice of him. So I wanted to make that mention tonight that Joanne Pepperell, his wife, he's got a son and daughter. So I just wanted to pass that on to members that have known Don for many, many years. He's been a great contributor on many fronts, but being legal counsel all those years was quite something for us, and then providing a place for us for our office for 29 years has been very generous. Wachiska is an independent chapter of the National Audubon, and we have 17 counties in our region, all of southeast Nebraska basically. You can follow us on our website or Facebook. The spring bird sale has started. It started February 1st and will run until March 7th. The order form is in the February Babbling Brook, and also in the newsletter, and then also Arliss brought some forms tonight. So if you didn't get to your copy or misplaced it, she does have a couple extra forms out there. So, or you can always call the office. So we've got those bird forms are out there, and that's till March 7th. And let's see, the pickup will be on March 15th and 16th. And this year we're going to pick up at the Wachiska office. We're not going to be at the old site over at the old post office. We're going to move it back to the Wachiska office, the garage on the south end of our building. So that's where the pickup will be. And I want to let everybody know that we're going to host the bird encounter at the Gear Library. Now, there's a little confusion because in the Babbling Brook, we had the Gear on one page and we had Walt Library on the other. Well, Walt's where it's been the last couple years, but it's actually Gear Library on South 56th Street. So that's a week from this Saturday, February 17th. It's going to be from 10:30 to 2, and there'll be a lot of activities, especially for little kids and families. So we were over there today, Tim and I and Patty Spitzer, and we went through and with the librarians and looked at the site. So that's going to be a good event. Little kids really love it. We have about 6 or 7 different activities, including making little bird cone or pine cone bird feeders and some things like that, use of binoculars and some stories and some music. So that, again, is February 17th, 10:30 to 2. And, again, no one's ever too old to come to an event like that. So if you want to see the little kids having fun, come on over. We're collecting new calendars at the office. As one of our members has been delivering them to recently paroled inmates and also inmates that are still incarcerated because they like to kind of keep track of their days, and so they provide those calendars. And so if you have some calendars piling up, like we get a lot of these calendars from all the different conservation groups, I know I get about 6 at home. So if you want to drop those by the office, we collect them. We just had a whole stack the other day we gave her, and we've already got a new stack. We've had a couple people come in. So if you have extra calendars and want to find a home for them, bring them on over, and we'll get them to a new home. Yes?
Only 2024.
Yes, only 2024, right. So if they're old calendars from 2023, no, we want the new calendars because we're giving them to these guys, or the day planners, the little day planners that is a calendar. They like those also. So whether it's a big or a small calendar, any kind, we'll take those. Our next board meeting's in 2 weeks on February 20th at 7 p.m., and it's via Zoom. And the meeting's open to the public, and if you're interested in zooming in, get a hold of Arliss or I, and we'll give you the link to it if you're interested in sitting through a board meeting. We'll take a few minutes here to see if there's any unusual bird sightings. Does anybody have anything they've seen or at their bird feeder, or has anybody been any place exotic lately? I know last month I talked about some of my travels, but does anybody have anything exciting in and around Lincoln? Yes?
I saw a bald eagle along the road between here and Palmyra.
Here in Palmyra, bald eagle. Good, okay. I talked to a gentleman from Unadilla this morning, and he's got a large red-tailed hawk nest, and he thinks at one time it might have been a bald eagle nest, but he said there have been a few bald eagles kind of checking it out again, so maybe they might nest in there. But he said there has been a nest down just outside of Unadilla a couple times in the last few years. I know over by Mahoney State Park, there's a couple in that stretch. So when you go to Omaha, be sure and look around the Platte River, watch the sky and those trees. There's one big bald eagle nest you can see on the north side of the bridge, great big structure. So yes, Lana. 20 bald eagles in Nebraska or over in Iowa? Between Plattsmouth and Glenwood, okay? And I hear there's a lot of trumpeter swans right now down at Los Hills, down at the Squaw Creek Refuge. So, okay. Anything else anybody want to mention tonight? Okay, well, this is a reminder too that tonight's presentation is going to be available on YouTube, and so you can check it out on YouTube if you missed it, or if you got some friends that weren't able to come tonight and you want to see it again, you can go to YouTube. And then also on our homepage is a link to last Saturday. Here in this room, we had a panel of 5 people on heat pumps. So if you're thinking about or need to get a new furnace or air conditioner, don't wait till last minute. Do your research ahead of time. But these heat pumps can save a lot of energy. So we had Kim Morrow, Councilman Bernie Shoby, Ken Harr, Mark Skolnick from LES, and Kurt Donaldson, who was the guy that got all the Lincoln Public Schools on the geothermal systems. Those guys were on a panel, and so it's a really nice panel. It's about an hour and a half. So if you have an hour and a half and you'd like to learn about heat pumps, go to our website, and from there you can click on that link, and you can learn all about heat pumps. Tonight, it's my pleasure to introduce tonight's speaker, Dr. Larkin Powell. Larkin grew up in southeastern Iowa and has worked for the last 22 years as a wildlife professor at the School of Natural Resources at the University of Nebraska. In August, he became the director for the School of Natural Resources. He enjoys playing the piano, photography, fishing, and poetry, so quite a Renaissance man, Larkin. Tonight, Larkin's going to talk about One Health student training in a contaminated landscape. He's going to be examining the Alt-In biofuel plant near Mead and its contamination in the area and ongoing research and monitoring. So I think we all saw a lot of that stuff go on with that over at Mead the last year here. So he's going to talk about that. So thank you, Larkin, for coming tonight, and we'll do questions afterwards. We'll bring a mic around so that we can put everybody on so that people at home can hear. So with that, I'll turn it over to Larkin.
Great. Thank you, Mark. Good evening, everybody. Sounds like the microphone's working okay. You got me all nervous. Usually if you mess up during a talk, as soon as it's over, it's just over. Now it's going to be on YouTube forever. So I am really glad to be here. I have stopped counting. This probably has to be like my 6th or 7th time talking to this group since I came as a fairly young wildlife biologist back in 2001. And it's always a pleasure to come chat with you. And it's a new role that I'm in now. This particular project that I'll share with you tonight, even though I'm 100% administration, and I've been told that my performance evaluations, they don't care how many manuscripts I publish now. I got to keep the school moving forward. I can publish if I want to, but that's not how they've been for 22 years. That's how I've been measured. But now it's the school. So we're going to focus on the school. But this is one project, even though I won't be having graduate students, this is one project, and I have a long-term painted turtle project out in western Nebraska near Ogallala and both of these involve undergraduate students. And so we'll talk more about that. And that's actually one of the ways that I want to focus the talk tonight, is to talk about how we use this experience as a training opportunity for students. I think I'll say one of the things that in my new role that I see is, I don't know if you agree with me, but it just seems like we're kind of on the threshold, or kind of environmental issues just keep kind of piling on, and maybe with a little more speed. And folks are worried about extreme weather events now, even if we don't want to talk about climate change. Extreme weather events. We've got farmers and ranchers that are very interested in that, and they come to the school for questions about that. So that's good to see that we can talk about climate in that way. There's things like the Alt-In that just show up, and water quality, and these things are important. But I also realize that those problems are not going to get solved before I retire. And so training the next generation to do those things is one of the most important things that I think that we can do. And that's I'm proud of the way that the school does that with our undergraduate programs. So, but I'll get started and talk about the topic tonight. But it is actually the topic tonight is how we use this experience for training. Let's see if I walk too far away from the thing. Let's see if what did I do? I'll do my little disclaimer while we're getting that Boy, it sure worked earlier, doesn't it? That's what happens when you test things out beforehand. My little disclaimer is that we're going to talk about toxicants and contaminants tonight, and I'm not that hasn't been my area of research in the past. I'm a population biologist. I like to look and see, especially how like habitat affects, and I've worked mostly with birds in Nebraska. What makes populations go up and down? What's their productivity rates? And so we monitor nests and that sort of thing. And so that's been my area of expertise, estimating survival rates, those sorts of things. But this thing came along, and just like I said, I was never going to work on wind turbines, and I was going to leave that to other people. Well, when they put wind turbines where my prairie chickens were, then I did a wind turbine project. So sometimes you have to kind of come back around. And so I actually did a contaminant study here but it was a group project. And so I rely totally on people from the University of Nebraska Medical Center and some other folks on our team to inform me about these sorts of things. How many of you have heard the Alt-N thing? Have you seen that in the news? And so some of you may have actually followed that even more than I have. And so you may actually know more details about this because you've read every single newspaper article. I know it's an incredibly interesting and unfortunate event. So that's my disclaimer to get that out of the way.
Well, the pointer worked.
It's not.
Can you advance on screen? Can you just advance them on screen and Larkin can tell you when to advance?
Is it if I just tell you to next slide? I wonder if you just have to like click on the PowerPoint just to kind of bring the PowerPoint back to being active. There we go.
There we go.
All right.
All right.
Now we're cooking with gas. Okay, so this is a slide on the left half. I just wanted to make the point. This is actually the team that has been doing the science. You'll notice my picture's not up there. I come into the project and I leave the project. But this is the core of the team. And these are people that have been doing, it's a multi-layered project after the event, and we'll talk about the event as it is, but when that opportunity came along to study that, we'll look at it in a positive way. When that opportunity came along, this team formed and is looking at human health concerns, soil and water potential contamination. So there's water and soils people up on that screen. There's people that are looking at insects. There's people that are looking at plants. And then there's people that are looking at the wildlife. And I'm part of the wildlife team, obviously, because we're going to talk about birds tonight. But those, and so there's Medical Center, University of Nebraska Medical Center people, and University of Nebraska Lincoln. And I think there's actually a couple of people from Creighton that are a part of that. So it's a diverse team, and they get together and talk. And then to add to that, Liz Van Wormer and Judy Wu Smart are right up in the middle of the, let's see if I can not mess everything up. So Judy is on the left, Liz is on the right here in that second row right in the middle of the pictures there. Judy's an entomologist. Liz is a One Health. She has a PhD and a vet degree. And so she's half in the vet school and half in our School of Natural Resources. And so those two decided to take advantage of this and create a training program for students. And so we have this internship program. The other way you can read that is that there wasn't a whole lot of money to hire graduate students, so we only had enough money to hire undergraduate students. That's another way you can interpret that. But it really has been a great experience, and we've actually been able to bring some graduate students into it that are being paid to do other things. So we're going to talk about sampling for bullfrogs, and we're going to talk about sampling for birds tonight. So the point of this first part is there's just a really big team that's been working on that. Well, now I'm stalled out again. There we go. All right, so just to kind of bring us all up to baseline, and if you have kind of forgotten what happened up at Meade, so I grew up on a farm in southern Iowa. My dad has corn and soybeans. He just retired about a year ago, so he doesn't have that anymore, but they still get planted on our farm. And you know, every corn seed is coated when it goes in the ground with some insecticides and fungicides and things to, the hope is the seed corn companies put that on there so that that seed can have the nicest entry into the world and not get eaten by bugs or not get destroyed by fungus. So there's still concerns. There have been concerns from the bird world around the country, and it's actually in some European countries you're not able to use these neonicotinoid insecticides that are part of that mixture that gets put on there. There's been concerns even about how much of that gets in the environment when you're placing that seed, you know, every few inches in the soil. What we would now call relatively a low level of those out in the landscape, because what we now know as relatively high level is what happened at Meade. Every year at the end of the season, after people have sold seed to people like my dad, the seed corn companies have leftover that they don't sell. And so now the question is, we can't use this because this isn't going to be it's not going to be fresh next year, and also we'll probably have new hybrids next year. So they've got to figure out how to dispose of it. And a lot of times they just will like put it in the landfill or do something with it. It's not safe. Everybody agrees it's not safe to feed to cattle, and so they don't take it. They can't take it and feed directly to cattle. But someone had a really brilliant idea, which turned out to not be such a brilliant idea, that if we just, that's still corn and we could make ethanol out of it, right? And so somewhere in some room, there was a group of people that mistakenly decided that this would be a great idea to bring all of the seed corn from around the country. Seed corn dealers from around the country would bring their stuff here to north of Lincoln, Nebraska, near Meade, at this ethanol plant, and it would be cheap because if my dad sells his corn to the ethanol plant that's near him in southern Iowa, they pay him for every bushel that they bring in. Well, this stuff is leftover stuff, and so it's going to be really cheap to accept, and it's also like a hazard essentially. And so we're actually doing you a service, right, to take it if we're the ethanol plant. Well, it sounded great, right? And they even had plans to take some of the materials and feed it to cattle or do some things to make soil treatments out of it, the leftover wet cake when it was done. And so it turned, and so you can see the pile here with all the colors in it. That's the coated seeds of all kinds of different colors that are piled up there. The plant was shut down in 2021. I'm going to show you some slides that Judy Wu Smart provided as part of our team, that she has some evidence that there was stuff going on in the 3-4 years before that, that there was some signals out there. And she was one of the first people to bring this to awareness. But because of the ethanol plant, there are the containment ponds where the effluent water comes off that comes out of the process. And then there is a product that comes out of it called wet cake, and that's what's up in this picture up here. It's after you ferment and create the ethanol, you've still got the corn, what's left of the corn that didn't get used up during that fermentation process. And so it's a waste product of that. Normally, when it's not treated seeds, like ranchers in the sandhills buy wet cake, and it's still high in protein, and you could feed your cattle with it. Prairie chickens ring the edge of these bunkers of ranchers in the sandhills that feed this to people in the wintertime because they love to eat this stuff. So wet cake has become a feed product, a protein product in other situations. Not used here. But so these piles of wet cake that they didn't know what to do with were building up, and then these ponds of effluent that turned out to be contaminated were there as well. This is Judy's little timeline starting in 2017. Remember, the plant was closed in 2021, and she has about 2 miles away from where the Alt-In was. It's still there, it's just not operational. The university, straight south of there, has the Eastern Nebraska Research, Extension, and Education Center. And so there's a large multi-square mile area directly south of that that my colleagues do at the College of Agricultural Sciences and Natural Resources do, soybean trials and grazing trials, and we have forestry trials that we do up there for the School of Natural Resources. There's a lot of different types of research and outreach display programs that are going on up there. And she has a bee lab on this. One of the parts of that is for the entomology department is a bee lab. And so they are testing different types of things for getting rid of pests of beehives and different things. Well, so she starts having colonies dying out fairly soon after they start to do this on that site. But of course she hasn't made the connection yet that it's what's going on at Meade at the Alt-In, but so you can kind of see her timeline there. And pretty soon she can't keep any bees alive at this site. And like something is going on, and she's got bees around the state, and there's something weird about this site because she doesn't have this problem at any of her other sites. And so she starts looking into things. There's and they start trying to raise some issues. There is some words up here. I don't have the pictures of this, but she actually had in 2018, there was a colleague of hers that took some milkweed leaves to look to see if plants were taking these products up. And sure enough, they had the plants in the leaves had what we call the metabolites. So you've got the initial insecticide that's on the corn, the seed corn, and as that gets taken up by either plants or animals, those cells metabolize it just like how we digest things, and it turns into simpler sugars and simpler proteins than what it was when we ate it. Well, it creates other chemical compounds, and we call those metabolites. But there's still things we can look for, right? It's not the original insecticide, but it's things that come from it. And so she had a colleague that showed, sure enough, it was in those. Well, she's an entomologist. That's disturbing to her because milkweeds, monarch butterflies, right? And so this is what we're a little worried about. She's a little worried about. So they met with folks. They kind of got pushed back a little bit. There's no problem. But she's very persistent, and so they kept talking to people. This is just an aerial view. So these are those ponds where that was. The site is here, and the ponds and the wet cake was being kept over here in this facility. There's some more wet cake there. And so here's some pictures that they were taking to show this too. That's not supposed to be green normally if you're just using regular corn. That's still got that residue on it. So the process of creating ethanol did not destroy the neonicotinoids. It didn't destroy the fungicides. It didn't. There was some initial thought by the folks that had this idea that the heat of that and the fermentation process would just take care of that, and it didn't. So it wasn't as simple as maybe they thought. So there's a lot of these wet cakes piles sitting around, and you can see here there's some rain, there's water that leaches off those during rains. And then this is the year that I got brought into this. In March, there was a big rainstorm, and these ponds were already having some issues, but they overflowed. And those ponds where this water that had come off the process for making ethanol that had contaminated water in them, we now know, they overflowed, and they went down a creek. And previously, remember, it's kind of been on this private site and hard to get any real evidence if you're not allowed to go into the site. But now you've got this effluent that's coming down this creek, and there's university employees that are watching it go past their field things. And these folks are scientists. And so they start to document things. Even though they're like agronomists and range biologists and things, but they're noticing that this kind of really stinks, and it's a foul color. And so there's documentation of how far down this creek it went. At the same time, I'll also mention, for the past couple years, that wet cake had one of the things that they decided, well, they can't feed it to cattle, but let's use it as a soil treatment. And so they were shipping that to areas around any farmer that wanted to take it. And of course, most of them had no clue what they were actually getting. And the idea was you stir that into your soil, and you'll increase the carbon, the organic material in your soil, and it'll be a soil improvement product. And it's free, by the way. We'll just give it to you. And so now you're starting to think like me, right? That, like, how are we even going to do? Normally we would take some sort of gradient approach, and we'd have like where the contamination was, and we could do different distances away from where that contamination is. Well, now we've spread that contamination around, right? It's gone down this creek, so now you've got this flow that now the contamination is not just in a spot, it's along this creek. It might have gone into the groundwater. And now also around the whole county, you've got different farms where wet cake has been taken. In some cases, it's been delivered and sat there in a pile for a couple months until the farmer decided to move it and disc it in. So it didn't get immediately incorporated into the soil. Just a mess. And it's a mess from an environmental standpoint, but it's also a mess to try to now figure out, like, how are we going to do our sampling design? Because in some cases, we don't even know where that wet cake went to. And we could have a field that we consider to be a control field where we don't think it was treated anywhere close to there, and then find out later that, sure enough, actually there was some put on close to that field. And so now it becomes a treatment field rather than a control field in the experiment. So it's just a mess all around. Just a little bit about how that you think about that contamination. This is from the normal situation where you just have like a coated seed, okay? And the ethanol plant isn't involved on this diagram, but it's kind of how it can get into the, whether it's sprayed or it's on the seed, how it can get into the food web. Of course, birds can eat it directly over there, so there could be, like, now you're thinking, okay, like that wet cake pile, a bird can land on it and eat that contaminated residue directly. It can seep down into the ground, go up inside of a plant. It's going to be diluted a little bit now, right, because it's gone through the ground and up into the plant. But then insects can pick up that from the plants as they are getting the pollen. And there's just, and then those insects are then eaten by birds. So you've got the robin over here that's eating the worm that used to be in the ground. And so it's an indirect path. So it starts to get into the food web. And there's lots of different ways that it could there's dust that could potentially blow. And so that soil can move to new places. This is a little complicated diagram, but it just shows what's going on here across the screen. from microbes, insects, pollinators, I guess, specifically the butterfly symbol, other types of invertebrates represented by the ants, aquatic invertebrates, oddly represented by a crab. Think of it as a crayfish in Nebraska, okay? And then you've got amphibians, reptiles, fish, birds, and mammals. And these circles, the farther out on the circle there's color, it means there's higher risk if it's out towards the edge of the circle. So if it's in toward the middle, not that much risk. And then the colors, green is always in the top left, brown is always down in the bottom right, blue is to the bottom left. That's blue for water, soil is brown, air is gray, and the plants are green. And so just to take mammals over here as an example, fairly like intermediate risk. It's up 2 levels. It's not at the farthest out. And it's actually from all 4 of those factors, mammals could potentially be at risk from getting it through the soil because they're down there digging through the soil and maybe burrowing in the soil. They're eating plants, they're drinking water, and they're breathing air. Some other things like the insect pollinators, like the butterfly, more of a risk from air and plants. So the point is, every critter and part of the ecosystem out there has a slightly different risk depending on its job in the ecosystem and how it might come in contact with it. So that's kind of interesting to think about how different things are maybe more at risk or less at risk than others. The fish, they're just fine as long as it doesn't get into the water. If it gets into the water, then they're in trouble too, right? So, so that's that. Now, to just kind of pause and think about, now we've got this perfect, what I call the perfect One Health issue here, or problem case. And we'll be talking about this for years at the university probably. This is Liz Van Wormer, my colleague, the one that's the vet and PhD that's back and forth between the vet school and the School of Natural Resources, and she's the head of Nebraska One Health. It's possible you've had her come talk to you here. There's a good chance. She talks to a lot of groups. The idea behind One Health is that people and animals and the environment are all linked. And so we can't just worry about human health without thinking about the environment and animals. You can't worry about livestock health without thinking about the environment and people around them. And there's a lot of examples of this, whether it's like, bird flu coming over and infecting, so the wild birds infecting chicken flocks. That's a great example of transmission across. And usually then, and people are at risk in that situation too. So a lot of complicated things. Liz runs a nice program. And so here's her description, and she and tonight's presentation is put together with some slides from our team to show you different parts of the project here. But this is another depiction of One Health. You've got the animals, the people, and the environment, and we have to work on a solution for everything if we want real health. So that's the idea. And then I told you that she and Judy developed this training program. And so this is some of the students that were out there. In this case, they're looking for bullfrogs as another type of thing to sample. Bullfrogs, why would you think that they might be chosen as if we were looking for an amphibian that we don't mind taking and putting in a blender, why are bullfrogs? I mean, eventually that's going to happen to some of them. It happens to the tadpoles. Why? Yeah, it's an invasive species. So they're one, if we're starting to talk about leopard frogs, well, now, okay, or, tiger salamanders or something, well, maybe let's not do that. But the bullfrogs are, and there's because they're invasive, there're just a lot of them out there in different places. So that was one of the reasons that they got chosen. And so we have a partner with the U.S. Geological Survey that is monitoring. She's actually based in California. They've been helping us because of the national prestige of this event. there's people from all over the country that are interested in helping with this. And so some of the samples have gone to California to look at. The biological samples have gone to a lab in California for their analysis. And so that's what they're out there looking for, especially tadpoles is what they're looking for. I think that's, so they've, as of, I think last fall, they'd collected 83 tadpoles, and they've sampled 43 of them so far. This is another site. This is actually Liz and a student. This is the creek that effluent went down, okay? And so this is the creek at kind of low tide. You can't actually, this is just what it looks like normally. It's moss, that's gunky stuff there. Isn't anything related to the waste that came down it, but the water that's left could have been residual. There could be groundwater, and the mud could have been contaminated. So they're looking for those things there. And obviously, I'm also looking for some blackbirds. We'll get to blackbirds in a second, but I'm going to be looking for some blackbirds that are nesting along this creek too. Dennis Ferraro is our herpetologist, so he's out there. And so we've got experts in the field. All the fields that we need are out there helping to train the students. They're learning to sample. They're learning about contamination. They're learning this theory of One Health and how that's important. And some of them have actually gone directly on to some graduate programs where they're doing that sort of thing. And that's fun to see coming out of this program. So he's trying to get a seine out there to see if he can catch some, see what kind of amphibians are hanging out in this pond. This is a student actually that's gone on to graduate school down in New Zealand. And she's doing a One Health project in New Zealand. She was a fisheries and wildlife student, Abby. But she was involved in the first year of the project. One of the problems we had sampling for bullfrogs the first summer or two was that there wasn't a lot of water out on the landscape. So this was a wetland that we had marked on the map, like we should go check this out for bullfrogs. We didn't find too many. It was pretty dry. It also made it a lot easier to walk across these wetlands to look for songbirds because they were still nesting in the vegetation. Why would we use red-winged blackbirds as the species to, they're not an invasive species, but if you have to choose a songbird that's out there, why would you choose red-winged blackbirds as one to take an egg from their nest? Yes, they're very abundant. Nobody's gonna, I actually, every time I take one away, I tell mother bird sitting over there that I'm very sorry. So we don't like to do that, right? But for the good of science here, one egg out of a few red-winged blackbird eggs near Meade, Nebraska is not going to affect the red-winged blackbird population that much. But that's exactly right. And in some cases, they're considered a pest species in grain and things, and people will net them and try to get them off crops. And it turned out I initially picked them because of the abundance thing. I didn't realize, even though I had studied red-winged blackbirds before in wetlands out in the rainwater basin, they nest in wetlands, they nest along streams, they nest in little plum brush, along fence lines. They're everywhere at that site. So if you're trying to get samples from across that site, near and far, red-winged blackbirds, good choice. One of the best choices of my career, probably. So that's,
Wear a helmet, though.
well, they do get a little frustrated with you. It does make it easier to find their nests because they kind of let you know when you're getting close. This is Ian, another student that's gone on. He's working at his PhD here at the University of Nebraska in biology. I've known Ian for several years, since he was an undergrad. He worked with turtles with me, and he was a graduate student that helped direct the students during the first year of this. You're aware, the female red-winged blackbird's on the left, the male is on the right, and so we're trying to find her nest, and the thing we're trying to sample is the eggs. Also, nobody cares that much about cowbird eggs, right? Because they're a parasite. So these cowbirds lay their eggs in other birds' nests. And so you can see the red-winged blackbirds are the blue eggs, the 3 here, light blue eggs that have the little splotchy coloring on them. I just think that's the most incredible. Every egg is different, right? Just slightly different. And then the cowbird egg has the little brown stipples on it. And that female cowbird, the light brown one, will go around, and there's been some studies that have counted up to like 35 to 40 eggs that one cowbird will lay out there in various birds. So they watch just like we watch to see where those nests are, and then they'll go in there when the host mother leaves. We call them the host because then they end up hosting this egg that is not theirs. So that we call the cowbirds a parasite, nest parasite. So we always take one cowbird egg too, just for good measure when we take, so those 2 species are what we're taking. And so eventually that egg, we put it in a cooler, gets put in a freezer, and eventually it gets put in a vial for sampling. And we collected 145 eggs the first summer. 100 of those-ish, 102 were red-winged blackbirds, 43 brown-headed cowbirds. And then the next summer we had a little shorter field season, 58. And then in 2023, we actually were back up, that's not on this slide, but we were back up around 100 and some eggs again. So it's fairly easy to find these nests, and they're out there on the landscape, and so it really made it a good sampling thing for us. But then those tubes are what gets sent to California to get tested. So far, as this says, we've actually only had 33 tested. It costs about $200 a test. So every one of these are not cheap tests, and the person is basically doing them gratis for us when she runs other things that she's got grant money for. And so it's taking a little longer to find things. But here's, I think that's actually Ian. You use behavioral skills to find these nests, so you just sit and watch. I'm sure some of you have done that too before, trying to figure out where mama bird's nesting. So you sit and watch, and when she comes down to the nest, you know that you've found it. That's actually this was the creek that effluent went down. Normally fairly dry creek, but one of our students here out, you can also stand off to the side with your binoculars and watch birds flying back and forth and see where they're dipping down, bringing insects back to the nest. And so a lot of these students, it's basically you're teaching them animal behavior while they're out there too. And so he had a great time. Another picture of a nest. Sometimes you find interesting things. This was when I went to do a nest check one time. Got a little snake in there. Now I'm actually blanking out. This is not a bull snake. Dennis is going to be very upset with me now because I've forgotten the type of snake that is. Anyway, it was actually in the act of eating an egg, and it before I got the picture taken, it got the egg back out of its mouth. But if you look real close, the egg is kind of shiny, and that's because it's got snake saliva on it. So you just never know what you're going to find out there in nature, right? We had the first summer. We also, because we really were not completely sure. We've had all these beehives that have just failed, and we didn't know what we were going to find because this was a really massive contamination event. And there was a possibility we were going to find none of these nests surviving. Birds laying eggs, it could have been like one of these DDT things where I had no idea what was going to happen, what we were going to find. And so we had a plan. We were also waiting. We would monitor the nests until they fledge and then take a tail feather out of the fledglings when they left the nest. And you can see their wing feathers here, and the tail feathers are just over my hand there, and there's just enough feather that you can get a little feather sample. And so we'd take one feather from those. Those have yet to be analyzed, and we haven't done that yet, but and obviously we have fewer feather samples than we have egg samples. And why do you think that is? Well, why is that? Okay, so we found the nest with the egg in it, and then we have to wait until it fledges to take the tail feather off the nestling. What could potentially happen in between there? Remember the snake? Or other predators could come along. So we actually only have about a 15% chance of having a successful nest. Or not we, the birds. We've done the data on this, and 15% of the blackbirds on our site had a successful nest. So that means we only get 15% of them. We only have a tail feather off of 15% of them because the rest of them got their eggs or the young nestlings eaten before we got to sample it. So we decided to focus on eggs because we had more of them. And I'll also, a little spoiler alert, the nest success ended up to not be different closer to the alt-in site or farther away. And we had birds right along that creek and right outside the gates to alt-in. There was a ditch nicely in a public space, so we could go down there in the road ditch and sample nests right by their gate and right by where the wet cake had been on one side. And they were hatching and producing young. Now, I'll show you. So it was not affecting them immediately in terms of it was not causing fatality of the young. I'm not going to do any more spoiler. There's some cute, I guess you call that cute. But when they get those feathers, they kind of grow in different spurts and stuff, and so all of a sudden they kind of look like aliens or something, you know. I just think little birds are the coolest. So that's a picture of us taking one of the. So this is where the middle of that bullseye is the alt-in site. Ian made this map, and all these little blue dots are where we sampled eggs from. So here's a road ditch, here's a road ditch, there's like a wetland there with a bunch of them, here's another wetland. Anything that's a line is probably either along, that's a creek, that's the creek that the effluent came out of. Here's our control sites. We'll come back to that in a little bit, but we went north. It was actually the in-laws of Liz Van Wormer, the investigator that's working with me, the One Health lady. She has in-laws that nested north, and the flow was to the south. So we're like, well, that's going to be our control site. We'll come back to that in a second. So we've got, and this just shows a little closer in. You know, we went around this square mile. Here's the site, and we went around the square mile. We found nests there. We've got that's the wetland, the creek coming out of it, different places. All this yellow is University of Nebraska, that facility there. And you can actually see like little interesting roads there. That's because that used to be a bomb-making facility. And so there's some really weird roads that used to be around the plants there. Of course, that led to groundwater contamination from that site too, right? So if you're up on this, you realize that these poor folks around Meade have now probably deserved Superfund status at least twice, which is not a good thing. Here's the students in the lab. They're taking those eggs and processing them, getting the samples ready. So here's some bullfrog results. So alt-in is in the middle of the map there. That's the facility. So we've got sites to the north that were the controls, and then 2 sites along that creek that came out of the site. So we don't have any bullfrogs. We could not find any bullfrog samples right up close to alt-in, which was unfortunate. There wasn't any standing water that we could get access to any closer than the ones you see on that. And so these, in the legend over here, all these really long names that I'm not going to try to pronounce, those are different metabolites of either the fungicides or the neonicotinoid insecticides that can be tested for. And what you can see is that in the control sites, except for one little one in the N3, there really was almost nothing there in the control sites. But in the stream, there were levels of concentration that were at least graphable, right? We can make a graph and it shows that there is some. The interesting thing is that we really don't know that much about these insecticides and fungicides in terms of, so I might say, well, you know, the yellow bars there, that's a pretty, yeah, it's not zero, right? And so what's your question? Is that like a bad level, right? Well, the thing is we don't know. And there has been not a lot of studies on like what levels in animals' body are close to lethal. What we do know is that bullfrogs make a decent thing. Bullfrog tadpoles are a decent thing to use as a sampling tool to look for the presence of these. We still have to learn more about like how to interpret the levels that are in their system. And we know from the fact that the bullfrogs were swimming around and we didn't find dead bullfrog tadpoles that they weren't causing lethal effects, but they were present in their system. Same thing with the birds. So here's some averages from our samples. All these, here's the map, and the way this map is made, the circles are bigger if the levels were higher. And so here's the Alt-N plant right there, and you can see that there's two really big circles, and that meant there were two samples of eggs right outside the gate that had really big samples. So blackbirds, red-winged blackbirds, and their eggs actually turned out to be a tool that we can use in the field. This is part of our project that hadn't been done before, to show that there had been some lab studies that showed that in eggs that were of birds that were fed these things, that those insecticides would be in the eggs. But we didn't know in the field if they would actually show up in the eggs. It was just we're like, let's try it, because some people in the lab have shown this. So this is actually the first time that this has been shown to work in the field, to sample an egg and use that to show where on the landscape this shows up. There was some moderate, well, actually fairly low levels. This is a little stream headed south. There were some low levels there. All of these are zeros. They're just eggs that we were sampled, but they're very small dots, and so they didn't have any neonicotinoid metabolites in them. And then this is the wet cake. So this is Liz's in-laws, what we had thought of as the control site. But it turned out that their neighbor told her in-laws after they found out that we were sampling up there, he said, you know, our common neighbor, or a third guy, had wet cake delivered and it sat there for a couple months before he moved it into the field. And so what we thought was our control site actually ended up to have a contaminant present, and sure enough, at the little pasture that had some blackbirds nesting in it, there were some low levels of contaminants in their eggs just from that wet cake being delivered up there. So again, that kind of just demonstrates this messy landscape of how those contaminants can get around. But this is going to be a nice publication for these undergraduates to publish, to show that you can do this, that you can sample. This is a different metabolite. It's a different metabolite than the one I just showed you, but it has very similar patterns. The ones right outside the Alt-N, in addition to up by the wet cake, actually had some in them. These small little dots they were zeros. So that's the highest samples are the ones closest to, and we also didn't know if it would spread, would it spread a mile away? Would it spread 6 miles away? We had no idea, so we were sampling a long ways away. But it seems to be fairly contained to things that are fairly directly close to where that is in the egg samples. So for the future, we're going to continue. The plan is to go out there again this summer and look to see like how long does this stay around, because there hasn't been any new contamination since 2021. So we'll keep doing some sampling to answer some questions there. We've got to get more of our samples that we already collected processed so that we can know the full story, because we've got a lot of eggs. We've tried to pick eggs that were from a variety of distances away from the site. When we knew we were going to get about 30 to 40 analyzed, we picked eggs that would be representative, but we've got more to sample. And we're grateful for the funding that we got from our Institute of Ag and Natural Resources. The legislators provided some funding to provide to the entire team to do some things. And the USGS, through their amphibian research program, provided some of the funds for the project too. Now I don't know, let me see if this is gonna do anything. If I can, nope. Can you click the screen? Can you click that video and just see if that'll click on the, so this is just an example if this works. There we go. So this is just the fun stuff you find when you're out sampling, right? We found a snake once in one nest I went up to. I mean, thank goodness we just have our cameras or our phones with us, right? That we can take video on. So here's a little nestling that's just coming out of the egg. And the thing I always forget, like how hard they struggle. I mean, just look at this little guy and how, or gal, and how much they're struggling to get out of that shell. And it's just exhausted, you know, and it still can't get rid of this thing on its head. So anyway, I really enjoy, you can tell I like getting out of the office, right? So getting to see a little bit of nature. Just a reminder of why we do the things we do. But I'll come back to end with and just say that I think one of the, it was a unfortunate opportunity that we had to study this. It's been fun to see how a team got put together to study it. And I think one of the best things that came out of it is this training for our students. And that's as from my, I'll step aside from my scientist role, and I'll just say from the director of the School of Natural Resources role, those are the kind of experiences that we want our students to be having. And because they're going to be the folks that help us solve these problems in the next couple decades. So that's what I'll end with there. Just don't ask me any contaminant questions here, but happy to take some
Bring
questions.
the microphone around. Here we go.
I understand, or I think I do anyway, that the companies that provided this contaminated seed are funding the cleanup process. Have they been willing, or have you asked them to help fund some of the research project?
It's a good question, and I don't know that. I don't know whether that was a part of the request to them. I think the request to them was to, dire, we want to get this cleaned up thing. But it sure would be, yes. I guess it wouldn't take compared to what they're spending to clean it up. I think like the first summer we had $20,000 to that was what paid for the 4 undergraduate student stipends and a little time for graduate students that summer, and a little bit of processing. So compared to the millions, yeah, we just need to get the right person in the right place to make that request, I guess. Good question.
Do we know what percent of the cake, wet cake, has been removed?
Well, so I think it went to a landfill type situation, if I remember right, and I believe, don't quote me now, but I think that most of it has now been taken off the site. So just don't quote me on that. But I believe that it's been mostly removed now.
Has the company that manufactures the neonicotinoids been helpful? Do they tell you how long this stuff is going to remain in the soil, whether it breaks down or it will last for a long time?
Yeah, it's a good, so those, yeah. I don't think the only companies that have really been involved with this are the companies that sold the seed that was on. There are, if you go into the scientific literature, there are studies that have been done because people have been worried about neonics for several years at this low level, right? And because even at low level, people were noticing that, like, you know, there were fewer insects along the fields and things that where this was applied. And so, and especially in Europe, there was a lot of studies over there that on insects. And so there's been a lot of laboratory studies. I don't know the answer to like how long it stays in the soil, but but some of that stuff has been worked out over the last decade and a half or so, just based on like laboratory stuff. But the direct answer is, I don't think anybody's reached out to those manufacturing companies directly in this.
What's being done with the excess contaminated corn now that,
Yeah, well, it's not being, I think the good thing is it's not being concentrated all in one place. So my understanding is that it's often just put in landfills in like, if my dad buys it from a seed dealer near Creston, Iowa, they're gonna take it to the Union County landfill. And that's, so, but it's, I mean, still, there's, you know, over time that builds up. Those are supposed to be sealed and all this stuff. But, so it's not in such a massive concentration. Yep.
Larkin, as you say, this has been studied now for 20 or 30 years, and we all have access to the same publications. Why are the Europeans so concerned, and we're kind of blowing it off and not worried too much?
Well, it's a good question. Different politics, I guess, is my answer. There certainly is a much stronger lobby here in the United States to maintain some practices that can help just really robustly. I think I'll also just say, and you can tell me if you think I'm right or not, there's a lot of agricultural policies that are different in the European Union, in the UK, because those countries are, have become, and have been for quite a while, like majority urban. And so there are, like let's take Nebraska. Again, I said I grew up on a farm in southern Iowa. The percent of people in Iowa and Nebraska that are farmers has been getting less and less all the time. We still have enough, and we still have enough that they have a more powerful voice compared to in the UK. And so I think we're able to, and those agri industries that are important industries and have a lot of power and voice, are able to still hold some sway here. My prediction would be in 15 years, you're going to see a similar change here as our society. I think we can see that the same dynamic playing out as and not to create a rural-urban divide here by saying this, but like when with nitrates in water, there's been a shift in the last few years as like cities have had to start paying to treat nitrates. Well, all of a sudden, nitrates are a bigger deal and in the news, right? And so now there's like economic reasons why nitrates that nitrogen fertilizer helps produce a lot of food and makes our food prices cheaper, right? But now there's an economics on the other side that's starting to be into that equation. And I think that's my really long answer for why it's like, at least my perspective, in Europe, densely populated countries and a lot less power of those same companies there relative because of the population density.
Are there other teams, other people doing research similar to yours out on this site because of this catastrophe?
Yeah, so there's a whole host of and things that I didn't show you tonight, but there's people sampling groundwater, there's people sampling the water in that same stream, there's people sampling plants, and Judy has people studying insects and bees still. And then there's a whole human component that's been of the medical center, folks that have been focused on mead. They just released a publication, for example, that showed that 30% of the people that provided samples in the town of Meade had traces of those same metabolites in their urine. So again, it's presence. There's no indication from that study that it's causing a problem, but it's showing, and people, you may have seen some articles in the paper, they had some studies of dust in people's homes, and they compared, they used Omaha as a control. The funny thing was, they actually found a few of them in the dust in Omaha too, but much higher levels in the dust in houses, because that was one of the concerns, was just, how the air might carry some of that over there. So they were doing the old, the white glove test, except they took it and then they put it in a bag. So.
Well, you might have answered this partially, but I was wondering, if Game and Parks has done any fish tissue samples, because, just the other day in this nice weather, I saw several people fishing along Salt Creek by Ashland, and that stream that you showed in your pictures, I think was Clear Creek. Looked like you were sampling some fish or something, and just downstream from where that draw is that came from the contaminated site, there was a lake on the neighbor's property that it went into, and if you took fish samples there, I bet you'd notice quite a bit of contamination.
Yeah, and there's that, because of that particular creek, there's not a lot of deep water in that. We even had trouble finding tadpoles, places that had enough water for tadpoles. But that one, where it all kind of ended up in the pond, I'm going to say 5 to 6 miles south of there, went through the creek and ended up in this little reservoir type pond on this guy's farm. He reported that fish died. So I think the newspaper carried a story that he said, I had fish die after that. I don't know if any of those got sampled or if that got reported in time to get any of those sampled. But so I guess the answer is I don't know for sure if there were any fish that got.
Yeah, I thought of these guys when I was seeing them fishing along there. I said, well, that pond that you're talking about overflows into Clear Creek. Clear Creek goes into Wahoo Creek. Wahoo Creek goes into Salt Creek. And
Yep.
they're
Eventually the
eating
Platte.
these fish, and I saw these silver carp jump out of the water, and I'm thinking they're filter feeders. The algae would be contaminated.
Yep.
They're sucking that in. And
Yep. No,
it's a vicious cycle.
yeah, I think you're ready to help us figure out some sampling. You're right on it though. I mean, this is how you figure out which one of those. So those Asian carp species would be a really good potential candidate, right?
We'll do one more question, and I'm sure that Larkin would be happy to stay afterwards and answer a few more.
Sure.
But we'll do one more here, and then we'll.
I thought it was still a Superfund site, right?
I don't know I know that it has had a level of status from the World War II contamination.
Okay.
I don't know if anything got declared because of this. I just don't know the answer to
Well,
that.
just follow up to that, I'm thinking of the Hippocratic Oath there with your undergrads out there. Are they taking precautions?
Well,
with
we actually,
exposure?
we talked about that a little bit the first year. So there were some, early on, we were using some masks. I think we were, yes, we were trying to, as they handled especially the aquatic samples, we were trying to use gloves. That was a question, yeah. I think when we started to see that stuff wasn't just dying all over the place, that we felt a little better about it. But a lot of sanitary things, and also remember, 2021 we were in still in COVID protocols too, so there was a lot of hand sanitizer that in huge buckets that was coming out that we were using. So I don't know. honestly, I mean, that's hand sanitizers for like, viruses or bacteria. It's not for chemicals. So, but we were at least trying to be sanitary. So.
Did they actually close in 2021 then? Because they wanted them to close and they didn't do it for what? 2 years?
I think it was sometime in 2021 that the plant actually stopped. So I think when we were out there, they were not actively creating new wet cake so by at least May and June, they were not active anymore.
Cattle across the street,
Yeah,
right? It got sold, right?
you're right.
To Texas
Yeah.
or something?
Right.
So nobody knows about that.
One more quick one over here.
Sure.
What laws were broken with what was going on there, and are there any policy changes as a result of that?
Yeah, so that's an interesting thing because of and I'm telling you all these things I'm not tonight. I'm not a legal scholar. But it was actually a lot less laws than you would think, given the magnitude of the situation. But there's some things that are not regulated, and so those chemicals are regulated in terms of how like workers handle them when they're working with them and putting them on the seeds, and they're regulated in terms of like the amount that you can put on the seed. But basically, once it leaves the bag and the farmer's, it's in the ground, it's not regulated anymore. And well, I guess even in the bag. So that I remember a conversation early on that essentially that they're in terms of like everything they did until you might say if you knew that stuff had these toxic chemicals on it, then you shouldn't have sold it to people that might have harmed their livestock, right? Like maybe, but it would just be a civil lawsuit. But there were no laws that were really broken in terms of the actual use of that in this ethanol process, because the regulation of it stopped back earlier in the process. And once that, that's my, you can tell I'm not a legal scholar.
So
That's
there
my,
were no laws broken because when it came out, it seemed like there was this sudden exposure of something that was being done that was against the law, and that they were.
well, I think the fact that there was this large amount of contamination out there, then, so now you've got it was probably shut down because of that, there was a they were contributing to this growing contamination. And so now you've got some potential like water quality laws that are, but even those, I think, I should just stop talking because but there was just a series of things where people were like, well, there's actually not really a law against that, but the fact that the contamination levels were so high, that's when the Department of Energy and the Environment came in eventually and said, we can't keep adding to that anymore.
It sounds awful.
Yeah, and so it's one of these, it's just a great example of like one of these things that's a gray area that doesn't get regulated to the point where something like this can happen. Yeah, no, I mean, it's a very, so here, I mean, this is like literally, we talked about the One Health, but later in textbooks, this is going to be like a chapter, a problem case. I mean, and we've got it 45 minutes away from Lincoln here. It's a very unique situation and one that nobody could have really predicted, but it's here and it's going to be analyzed and looked at in terms of the law and the effects and what happens for years.
Well, let's thank Larkin for coming tonight. And I'm sure you'll stick around and answer a few more questions. We do have some brownies and some hot tea and water out in the lobby, so feel free to stay for a few minutes and enjoy your company and visiting with folks. So thanks for coming. If you didn't sign in, be sure and sign in on your way out. We try to keep track for numbers to report on our annual reports for people that attend events like this. So if you didn't sign in, go ahead and sign in. And thanks again for coming tonight.